Okay, so each year I have a similar title, just the number changes. 100,000, 200,000, 250,000. Maybe next year will be on 1,000,000 because things are really speeding up. So the whole story started 2007 when Pauline developed her first high throughput glycomic assay. And then I had a fantastic PhD student that called Anna Knežević, then now it's Anna Moženić. And she was halfway through her PhD, and I said, you know, Anna, do you want to give up everything you did so far and try something completely new? Try analyzing 1,000 glycomes. Because my previous PhD student did a PhD on 3 patients and 3 controls. So we said, let's move from 3 to 1,000. And then Anna actually went to Dublin. Pauline moved to Dublin then. Half of the cohort was analyzed in Dublin, half of the cohort in Zagreb, and then we published this first high-throughput glycomics paper, variability, heritability, and environmental determinants of the human plasma and glycome. And after that, we got many grants. So we got 6 FP7, 6 Horizons, 9 Horizon Europe. We have 9 ongoing Horizons, including the ERC project. Fortunately, most of them I'm not the one writing anymore. It's the team who is taking the effort. And actually, we submitted something like 10 grant applications in the last month, and we are submitting, I believe, 4 additional on Wednesday. So there are some people writing in the background. But we did manage to get a lot of money and analyze a lot of samples. And we did a strategic decision not to analyze any samples, but to look for the best researchers all around the world who do some really cool studies, who have great cohorts with the great data, add glycomics to their studies, and then try to understand a little bit more about protein glycosylation. And in the last 15 years, we analyzed something like 80% of the glycomes that were published. So we really kind of industrialized glycan analysis. And of course, this resulted in many interesting papers. And I think we are kind of switching the perspective on glycosylation, because for a long time we were thinking glycans are receptors for lectins. So you have a lectin will bind the glycan, and then something will happen. But this is actually just a tiny little part of glycosylation. We should think about the glycans as a part of protein structure. But a part of the protein structure is a polypeptide. This is encoded by gene. This is evolutionary wisdom of billions of years. You don't want to change it. You keep it and you give it to your children the way it is. But the other part of a glycan structure is encoded by a complex network of genes, which makes it very variable and very dynamic, but it's still a part of a protein function, protein structure, and a part of the protein function. So we can actually change our proteins literally by the way we think. So if we think in a different way, our proteins can be different. So from functional aspect, alternative glycosylation, adding a different glycan to the same glycosylation site, is the same thing as a coding mutation. So geneticists know that the coding mutation can kill you. You have a wrong nucleotide, wrong amino acid, the protein doesn't work. The same thing can happen with alternative glycosylation, but not by a single mutation, but this complex network. And this is a network of genes which regulate IgG glycosylation. I will mention it later. But from evolutional aspect or from informational aspect, glycome composition is inherited as a complex trait. And this enables diversity without the risk to genetic heritage. So we can change without changing our genes, without losing all the knowledge from the past millennia. And just earlier this year managed to publish a paper which was pretty convincing in showing that the evolution of glycoproteins peaked when the life was becoming multicellular. So the glycosylation has archaeal origin. So archaea invented and glycosylation, so before the bacteria and eukaryotes. But the peak of glycosylation genes actually evolved when there was grouping of cells in the colonies, when there was multicellularity, different tissues. So literally, the invention of glycosylation enabled life to become multicellular. And it's also enabling diversity. This is the cool study we did with Grant Morahan from the University of Western Australia. So he generated a collaborative cross-cohort of mice. Started with 8 mice, as genetically diverse as possible, crossed them through 3 generations, and then bred them into clones, into strains, which are as close to clone as we can get. And some of these strains became completely separate from the others without a single mutation. So this is just a reshuffling of allelic variant, making mice different from other mice. And we see the same thing in humans. The main reason why we are all not the same is because we glycosylate proteins in different ways. And this goes all the way from the blood groups. Blood groups are the glycans by chemistry. And this is affected by our genes, by epigenetic relation of these genes, and by environment. But the complexity of glycosylation makes it challenging for many scientists to get into this field, because there can be multiple glycosylation sites on the single protein. On each site can be a different glycans. It's really complicated to understand. So we really need high throughput glycomics to be able to understand glycosylation. And something I keep repeating for the last 20 years is that small studies generate irreproducible data. I generally do not take photos with people. I don't have many idols in this world, but this guy is one of them. John Ioannidis, probably one of the largest geniuses in science. He published more than 20 years ago that something like 90% of published literature is simply wrong, because people don't use proper statistics and they do small studies. And we do have this problem in a glyco field. So there was a comparison of IgG glycosylation analysis in 76 laboratories all around the world. 106 data sets were generated, published in Molecular Seroproteomics a couple of years ago, and the results were actually horrifying. Some of the labs had something like 100% CV values. So definitely we need better analytics globally. And for that, we need standards. We need common analytical standards which will help us understand when something went wrong. This is an example of our studies. One standard in 6 different 96-well plates. And what we can see here in plates 3 and 4, the glycome was really different. Some of the glycans went 20–30% off. And this is not biology. This is standard. So this is just analytics. So occasionally, our glycan analytics generates wrong data. But we have to monitor it, we have to identify it, and then we can correct it. And this is what we are doing now. We are developing a glycan analysis kit, which is actually already on the market. It's a CE-IVDR, which will have a common standards, which you can use to monitor your results and identify a problem, and then fix it if needed. Because you can either repeat it, so this is a repeated analysis of the same plates. This was just something went wrong in our dozens of steps we have to do in a glycan analysis. You fix it, then it's again. Or you can even use more advanced batch correction tools to kind of fix it just mathematically. What is shown here is something like 496 well plate assays we did for a glycan age. Before batch correction, some glycans behave pretty well even before the batch corrections, but sometimes you see things going off. After batch correction, it is fine. So I'm really happy that today we have a system which delivers analytical accuracy which exceeds most routine diagnostic assays. We have a dedicated glycan age profiler produced by the Nanjing Superiors. It's actually in the box in the back. It will be opened and put up there to see, and there will be some people from Superiors that we could talk about. We are developing CE-IVDR compliant chemical kits, meaning they have a regulatory approval to be used in the clinics. And then we have CVs which are really good. We aim to go below 1% eventually, because if you want to do proper analytics in a hospital, the results have to be reliable. Another big challenge we have in the field is bioinformatic tools. this is another horror story from the glyco field. This was raw data was generated in a single lab, on a single machine. And then it was sent to the different labs, in this case it was 22 labs, just to analyze the data using their software. And different labs identified between 99 and 168 I think the glycoproteins that were identified. The ratios, quantitative, were all around the plate. So even if the data is generated on a single machine, you just send it to the different softwares we have, and we get a completely different data. So we do need to standardize, we have to validate, we have to introduce external controls if we want to move into clinics. And I think this is something what we here should be working on. So, with the tools that we have already, which are most of the research tools, what did we learn? So, one of the first things we learned is that glycosylation is not regulated by glycosyl transferase. Full stop. We need glycosyl transferase. In the same way we need ribosome to make protein. But you do not regulate protein expression by changing the level of ribosomes. The glycosyl transferases are tools, they deliver glycosylation. And if we talk about the network of genes which regulate IgG glycosylation, yes, we have them here. But actually the decision makers are some other genes. And we did a lot of GWAS studies. Yuri will talk later about some of them. We generated a lot of hypotheses which genes are involved. But the GWAS is a hypothesis generating tool. Then we have to validate all these ideas. And this is something we are doing in collaboration with Lauc and her team. We'll have Annika talking later today about the most recent progress in how we are actually validating, functionally validating individual GWAS hits to see what these genes actually do. Because what we learned from genetics is that the genes which regulate protein glycosylation are known risk factors for literally hundreds of different phenotypes. So they are important for human biology. We just have to understand why. We also know that glycans change in literally all diseases. It's excellent review from Sophia in the Biotech Advances is that showing that glycans change in the majority of diseases, sometimes in a specific way, sometimes in a very similar way in the different diseases. And very often, these changes happen up to a decade before people get diagnosed. Because, you know, diagnosis is an endpoint. Something stops working, you go to doctor and ask for help. Molecular processes leading to diagnosis start much earlier. And we have a number of very prominent papers showing glycans change before you develop diabetes, before you have a heart attack or stroke, before you get complications, hypertension, whatever, RA, COVID, and so on. And this is not surprising because glycans are literally one of the key regulators of inflammation. Inflammation is a very complex term, but glycans have multiple roles. And actually, Claudio Franceschi, 25 years ago, claimed the term inflammaging. Inflammation leading to aging, where also glycosylation is very important. And I can announce that next June in Dubrovnik, we'll have a workshop of 25 years of inflammaging with Claudio and me organizing it. So keep in touch, follow our social networks, and join us if you're interested in glycosylation and inflammation. So glycans are the ultimate layer of molecular complexity. They integrate genetic, epigenetic, and environmental factors. Information collected from our prenatal life is encoded in our epigenome. Our lifestyle, our microbiome, the way we think integrates and changes glycan structures. And we know on IgG, this is literally changing IgG from pro-inflammatory to anti-inflammatory. We don't know all the details, but we know they're very important. And today, we can reliably quantify it. And we are trying to push it into the clinics, but going an easy way, so not going into hardcore diagnostics immediately, but first addressing the wellness and general health and wellness market, because what we know, that IgG glycans change significantly with age. as we are getting older, they're becoming different. And this was first shown in '85, and now we've shown it multiple times. Glycans change with age. And when we published this first time, and it was 12 years ago, the editor said, "Are glycans the holy grail of biomarkers of aging?" I believe that we are convincing the aging field that they are, and I hope in a few years we'll definitely convince them. Because we do have this commercial test available called the glycan age test, where the glycan age doesn't mean age measured by glycans, but the glycan age means the new age. There was a Bronze Age, Stone Age, and now it's a glycan age. It's not the DNA and protein age anymore. Quantifying IgG glycans and monitoring on a personal level how we are progressing through our life trajectory. And of course, I'm inventor of the test, I'm co-founder of the company, I'm conflicted. So don't take my word on that, look at the data we are generating. And the reason why we are kind of starting to dominate the aging field is that we invested sufficient efforts in analytical accuracy. So there's a lot of research about epigenetic age. And there will be a lot of research. It's fascinating how epigenetics is regulating aging. But when you come to an individual patient, epigenetics still does not have a tool to monitor individual patient because they use epic chips. Epic chips are a research tool. And this is a study we did with Alton Scientific and Jamie Haywood. These are his samples. 10 samples taken on 2 days with epigenetic age going 5 years up and 5 years down. And expected change of any kind of intervention is maybe 1 year up or down. So analytically, this is completely useless on a single patient. Of course, if you do 1,000 patients, measurement error will level out. You see the biology behind it, but to monitor an individual patient, you cannot use epigenetics today. This is how the glycans looked in exactly the same samples. And today, there are over 2,000 clinics which already use glycan age, and even the most elite one, the La Prairie, is using glycans as kind of their ways to promote that they are kind of better than others. Glycans are hidden signals of your health. And this information can really be useful. For example, this is my own IgG glycan composition. And the first thing which I think is really fascinating, this was not analyzed as a batch. This is a sample I sent to the lab every few months. And these are percentage of G0, G1, G2, salic acid bisecting. And literally the difference is very, very small. So we quantify glycans very good. And then you see something changing. And even it's a slight change, it's a consistent change. And I didn't like it because this is the glycan which is kind of protecting from heart attack and stroke, I will show it later. This is the main pro-inflammatory. So there was some kind of chronic inflammation in me I was not aware of. And I did all the checks, I couldn't find what was going on. And eventually I found the cryptic root canal infection, which is kind of, stupid thing, bacteria growing here, but it's a known risk factor for heart attack, for example. So this test enabled me to identify something which I couldn't see in a mirror, which I cannot detect in a routine health checkups. I fixed it, and it got better. And in many diseases, glycans resemble accelerated aging. So this effect of aging, and this affects of dozens of diseases. We have a nice review from 2 years ago, and pretty soon we'll have another very nice paper putting this all together, because what we are now trying to say is, okay, if IgG glycan changes in the same way in many different diseases, it means it changes in the same way if you are healthy. So the opposite of different diseases is being healthy. So now we are claiming that the IgG glycome, so-called young IgG glycome, is a biomarker of resilience, of you staying healthy. And this is something which is becoming really popular. There was an editorial in Nature Medicine by their chief editor saying, let's not focus on individual diseases because they are not well defined anyway. Let's try to see what is making people healthy, what is helping us to cope with all the challenges we have in our life. And glycans can definitely help you. So this is unpublished data. It will be published literally, Annika, in 2 weeks.
Hopefully.
So we have a paper written, we are just finishing up, we'll post it in MedRHab in 2 weeks. This is over 5,000 people from the German chronic kidney disease cohort, which were sampled at different age, followed up for the next decade, and this was their glycan age. Blue ones are still alive, red ones died. Meaning, if you had a higher glycan age at the time of sampling, your risk of dying is much higher. And we are literally publishing 3 different cohorts. We didn't want to go early with such a strong claims because we wanted to have multiple replications. We have 3 independent studies, one with the Karolinska, one with this German chronic kidney disease cohort, and one with the Scottish guys, and they all showed the same thing. When we tried to quantify it, and this was done on the Croatian cohorts, each increase in the glycan age is increasing the hazard ratio of dying in the next 5 to 7 years for average of 4%. More in men than in women. So people having 20 years higher glycan age, which there are people having more than 20 years higher or lower glycan age, this means something like 200% higher or lower risk of dying in the next period. So I do believe that this biomarker will become very popular soon. Interesting thing is, it does seem to quantify risk of dying separately for other known risk factors. For example, this was study done with the Peter Joshi. He developed mortality risk index based on the blood biomarkers. So routine blood biochemistry, everything which is done routedly on a UK biobank, when you put it all together, it can predict mortality. This is the effect of this kind of dedicated mortality test. This is the prediction of glycans. So the IgG glycans themselves are better than this everything together, but interestingly, it is highly additive. So there are 2 different aspects of mortality risk which can be caught by kind of a classical risk markers which are used today in the clinics and glycans. Also, we compared these different aging clocks, most of the epigenetic aging clocks, and generally they do not correlate. So of course, they do correlate as a measure because they correlate with aging. But when you look at acceleration, and this was published a couple of years ago, there's nearly no correlation between the glycan age and different epigenetic clocks. And also we have this replication study. As I said, we don't like to publish anything before it's replicated several times. And clocks correlate, but correlation of acceleration is very low. So epigenetic aging, routine blood biomarkers, they're catching different aspects of aging than what IgG glycans do. And aging doesn't seem to be linear. And there are a couple of papers suggesting that aging happens in some kind of bursts in different periods of time. What we have found, for example, in the Down syndrome, where we do know that Down syndrome is associated with accelerated aging when you look at the people in the single time point. But when you look longitudinally, actually the pace of aging of people with the Down syndrome is the same. They just have a rapid acceleration in childhood, and then they keep with the same pace. And just a few days ago, there was an update of this very fascinating paper called The Rhythm of Aging, Stability and Drift in the Human Senescence. It's still unpublished, it's only on, it's posted in Archive, but it does seem that with the progress of civilization, we did not change the pace of aging, but we actually kind of shifted this rapid burst of aging later in life. So it might be possible that we can shift it even more and prolong the lifespan. Because I think this is the most interesting question for many people, especially as they are getting older, is can we revert this glycan aging? Can we slow down or even go backwards? And we did a lot of studies. These are mostly small studies because it's very hard to do intervention studies in humans. But we know that the physical activity, diet interventions, hormones change IgG glycome. Of course, anti-inflammatory biologicals do the same things. But there are some kind of also exotic things like exosome fraction of porcine plasma, which does seem to have a strong effect. So something which repeatedly works in all the studies is caloric restriction. If we eat less, we will live longer. But I think this is so clear only because we all eat too much. So majority of people eat too much, so if they start eating less, things get better. And we did a cool study together with Luigi Ferrucci, who is the director of the National Institute of Aging, showing that even after 2 years of caloric restriction, you still see beneficial effects on the IgG glycome. And something which is really intriguing and the most fascinating thing we discovered in the maybe last couple of months is fasting. So eating nothing or very little for a few days can change your IgG glycome in 3 days. I didn't believe it when you saw it first time. I didn't believe it when you saw it second time in a cohort. Then actually I did it myself. And after 3 days of not eating, my IgG glycome is getting better. So there is something fascinating in fasting. We are doing a lot of research in this direction now. Ivan will have a presentation also in this conference, so you'll hear some of the recent data. But the moment you start doing things like changing your diet in a kind of a normal way, then there is no magic diet. And diet is more like a religion. People have their diets, they believe they are fantastic, and probably they are for them, because we are all different. So this was 1,000 people on 5 different diets, 200 people in each of them, followed for 6 months. And on each of the diets, including the high glycemic index diet, some people got better, some people got worse. So we are all different. We have to identify diets which work for ourselves, and to be able to do it, we have to monitor it in some way. Good thing is, whenever we start living healthier, things improve. This is people losing weight in their 30s, 40s, 50s. It's never too late to take care of your health. Physical activity is extremely important, but don't overdo it. So this is the typical Croatian people. We are generally couch potatoes, overweight, and some of us are more active, and they are younger. These are people who are regularly physically active. But professional athletes are actually worse than the couch potatoes. So if you overdo it, you're harming your body. But if you take a couch potato and try to force him to exercise, it takes a long time to see positive effects. We had 1,000 people exercising. They got worse in 3 months. They got worse in 6 months. Only after 9 months, some people started to get better. So physical activity takes time to have beneficial effects. Too much physical activity is actually promoting inflammation. We don't want to be physically active. We don't want to be hungry. We want to have a magic pill. We want to have a pill which will make us live longer. Well, if you talk about IgG glycome, you take infliximab, which is anti-inflammatory biological, your IgG glycome gets better. Side effect is cancer, so I don't suggest it as a way to promote longevity. But even infliximab did not change IgG glycome in some people. So we are all not the same. We don't respond the same way in this, even to such a strong drugs like infliximab. Another drug which seems to be doing wonders, especially in women in perimenopause, is hormone replacement. This was a cool study with chemical-induced menopause, supplementation with estradiol, or placebo group aged 30% in 2 months. Estradiol was sufficient to prevent it. Also, when you look at the population cohort, so generally women are protected from glycan aging in their reproductive age. With the rapid acceleration around the age of perimenopause, we saw it in all cohorts. So it's kind of slower, then rapid, and then slower again. And this is something which can be improved with the hormone replacement therapy. The problem with HRT is that first it's hard to identify the problem, because if you try to quantify hormones in perimenopause, they go up and down within a few days. So you cannot actually measure the hormone level and learn something. But this is where IgG glycome can also help, because it's strongly affected by estrogen, and it's integrating estrogen concentrations in the last couple of weeks. So actually, it's something like HbA1c for glucose. And we are also trying to position it in the clinics in that way. And for HRT, you can then monitor individual level how things are happening. You see some people responding, some people not responding again. But glycans will not be a universal solution, but it will be one of the biomarkers helping clinicians to make decisions. We more or less tested every longevity intervention that exists. For example, Tim and I even recorded Instagram Live saying that metformin helped him. But when we did the placebo-controlled randomized trial, only a single healthy individual actually benefited from metformin. Young and healthy, don't take metformin. Different anti-diabetic drugs, some respond, some don't respond. Some magic things like a therapeutic plasma exchange seem to be working. Exosome fractions seem to be working. So we have to find tools to help people stay healthy, not end up in a disease. And at least what you can do now, you can monitor. There's another famous people, my daughter, CEO of Glycanage, is also here. So she had a very good glycanage, then it got worse. But she identified it and fixed it. And then, again, it got worse, identified and fixed it. Because we know this is leading to disease. And if you don't know you have this problem, you're not able to fix it. It's a lot of data, but there's a lot of people behind it. So I have something like 60 researchers in my team, 30 people in Glycanage trying to translate it, and I hope we'll really bring glycans to the clinics within the next few years. Thank you. I think I'll use my time for questions.